The third portion of notes is about proteins and nucleic acids. So proteins are a major constituent of living things. They account for more than 50% of the dry mass of most cells, which means that most organisms are at least 50% protein. There are lots of different kinds of proteins that have all kinds of different functions, like support and communication, movement, transport, defense against disease and foreign substances. And so we'll talk on also enzymes and various things like that. So we'll talk about some of these functions along the way. There are, on the next slides, there are some descriptions of the different functions that you will be responsible for. So enzymatic proteins accelerate chemical reactions. They are biological catalysts, as you learned about before. This includes things like digestive enzymes that break down molecules like sucrose into its component parts like glucose and fructose. There are storage proteins that store amino acids for developing embryos and young offspring. Casein, which is the milk protein, is the source of amino acids for baby mammals. The plants have storage proteins in their seeds. Ovalbumin is the major component of egg white, which is the source of amino acids for the developing embryo inside the egg. Defensive proteins like antibodies, these Y-shaped molecules here in purple, protect you against disease by inactivating and destroying viruses and bacteria. We'll talk more about how those function when we talk about immune systems later on. Transport proteins like hemoglobin and transport proteins through cell membranes like this one right here for facilitated diffusion allow for the movement of molecules from one place to another. Some function like hemoglobin in a cell like the red blood cell. Others are for facilitated diffusion or active transport of some kind like a sodium-potassium pump. Hormonal proteins like insulin coordinate body's activities. So insulin is the hormone that allows cells to take up excess sugar, and so when you have a high blood sugar concentration like you see here, the insulin enables the cells to take up extra sugar, and that reduces the blood sugar to the normal level. And it's got a partner hormone, glucagon, which when the blood sugar falls too low causes the liver to release glycogen, which will or break down glycogen into glucose, and that'll increase the blood sugar until it reaches the set point. Contractile proteins and other motor proteins involved for movement. Examples are in the muscle tissue. The striations here in the skeletal muscle tissue are made of alternating fibers of actin and myosin, which enable your muscles to contract and then release. There are other kinds of motor proteins for other kinds of movements like the cilia and flagella as shown here. Receptor proteins are function in the responses of cells to chemical stimuli. So here you see a neuromuscular junction. So you've got the neurotransmitter molecules that are being released here, and there are receptors on the muscle cell or on the cell that the nerve is near, and that will cause that reaction to be transmitted to the cell. Structural proteins like keratin, which is found in hair, nails, and things like that, function for support. We've also got things like silk fibers made by insects, collagen and elastin that are found in connective tissue in mammals will provide structure and support for the tissues of mammals. Enzymes, in particular, we'll spend some time talking about enzymes. They act as catalysts to speed up chemical reactions, and the great thing about enzymes is they can do this over and over and over again without being changed themselves, and that's really necessary to speed up the reactions that are required for living things. So the unbranched chain of amino acids that makes up the protein are called polypeptides. They are just the chain of amino acids. They use the same amino acids, the same 20 that we have available in all living things. And then to become a protein rather than a polypeptide, the polypeptide has to be put together in a form that is biologically active. Sometimes it requires just bending or folding in a certain way. Other times it requires being associated with other polypeptide chains. So we're going to differentiate between the polypeptide and the protein by talking about the protein being the functional molecule rather than just the bare bones link together amino acids. Amino acids are the monomers of proteins. They are organic molecules that have a carboxyl group on one side and an amino group on the other side. There are 20 different amino acids that vary from this basic structure by having different R groups or side chains that can be added. There are some of the amino acids that have 2 side chains attached to this alpha carbon here. And the presence of those side chains or those R groups are what give the amino acids their different structure, their different properties. And when they go together in the polypeptide chain, they can affect the shape of the molecule by their interactions with each other. The amino acids are linked together by a type of bond called a peptide bond. This is similar to the glycosidic linkage, except it is between the carboxyl group of one amino acid and the amino group of another. The polypeptide, remember, is the polymer of amino acids, and that can range in length from a few amino acids to hundreds or thousands of the amino acids put together. The sequence of bases, or the sequence of amino acids in the polypeptide will make the polypeptide chain. One end of the polypeptide chain will have a carboxyl end, that's called the C terminus, and the other end will have an amino on the end, and that's the N terminus. And so that's going to be unique to each polypeptide. Here we have the formation of a polypeptide. We have 2 here that are linked together in a peptide bond. Notice peptide bond is between the carbon with a carbonyl group and the nitrogen of an amino group. So the carbonyl group is what's left over from the carboxyl group, and it forms a bond this way. So you lose the hydroxyl part of the carboxyl group and one of the hydrogens from the amino group. Those two go together to form a water molecule, and that leaves the carbon and nitrogen available to bond together in the special peptide bond. So here we have a polypeptide made of 3 amino acids. We have the N terminus, the amino end of the chain, and the carboxyl end, or the C terminus of the chain, and the different side chains attached to the different amino acids. For every two amino acids that join together, there will be 1 peptide bond. 3, there will be 2. 4, there will be 3, and so forth. So to become a functional protein, you have to have 1 or more polypeptides that are precisely shaped for the specific purpose that they are there to fill. Sometimes they're twisted or folded or coiled, and that depends on the sequence of amino acids. That's going to determine that 3-dimensional structure. And then the structure is going to determine its function. Proteins are very specific and they have to have a specific shape in order to function properly. The primary structure of a protein is its sequence of amino acids, of course, which is coded for by the DNA, as we've talked about in your introductory biology class. The secondary structure is composed of coils or folds in the polypeptide chain, and most proteins have at least primary and secondary structure. They all have primary structure, of course. The tertiary level of structure is formed by interactions among the side chains or R groups, because some of them may be polar, some of them may be hydrophobic or hydrophilic, and they're going to form various kinds of interactions with each other, like van der Waals forces and hydrogen bonds and so forth. The quaternary structure occurs when you've got a protein that's made up of multiple polypeptide chains that fit together in a certain way. And so we're going to look at each one of these. So the primary structure is just like the letters in a word, and it's going to be determined by the genes that code for the sequence of amino acids in a protein. And then the coils and folds of the secondary structure are going to depend on hydrogen bonding that occurs between the parts of the polypeptide backbone. The 2 kinds of secondary structure are a coiled form called an alpha helix and a folded structure called a beta pleated sheet. The alpha helix forms because there are hydrogen bonding that occur between the hydrogens here attached to part of the chain and the oxygens that are also part of the chain. And so you end up with those between the backbone parts, that those hydrogen bonds that occur there cause the coiling to occur. In the beta-pleated sheet, you've got interactions in a different way, but they're still formed by hydrogen bonding. And so the beta strand is usually shown as a flat arrow like this that shows the folding with an arrow pointing toward the carboxyl end. And you have interactions between the chains that fold in the same way like that. So it's still caused by hydrogen bonding. The tertiary structure is caused by interactions between the R groups rather than the backbone. And so you can have a lot of different kinds of interactions like hydrogen bonds, ionic bonding, hydrophobic interactions, van der Waals interactions. Sometimes there are polar parts of these side groups that repel each other or are attracted to each other, and all of those things can cause variations in the shape. There are also strong covalent bonds that can occur called disulfide bridges, and that gives a lot of reinforcement to protein structure. So here we have various kinds of bonding, hydrogen bonding between a hydroxyl group and a carbonyl group here. We have hydrophobic actions. All of these methyl groups here are hydrophobic, so they're going to have a tendency to stay together away from more hydrophilic parts of the molecule. We have ionic bonding that can occur between an amino group with an extra hydrogen and a carboxyl group that's lost its hydrogen to the amino group there. Disulfide bridges and so forth. So all of these things can affect the overall structure, the tertiary level of structure of the protein. And then finally, the quaternary structure occurs when you have two or more polypeptide chains that together form one macromolecule. An example of a quaternary structure would be the collagen fiber. This is three polypeptides that are kind of coiled together like a rope, and just as that happens with the smaller fibers in the rope, as you coil them together, it gives a lot of strength to the fiber. Hemoglobin is another example of a protein that you know something about that is a globular protein, and it's made of 4 different polypeptides, 2 alpha chains and 2 beta chains. So here we can see the alpha subunits in the dark blue and the beta subunits in the lighter blue. Each one has an associated heme section that has an iron molecule in there, and that's what gives the overall hemoglobin, globular hemoglobin molecule, its affinity for oxygen is the iron. So the physical and chemical conditions in which the protein is found can also affect its structure. If you change the pH or the salinity or the temperature or other kinds of environmental factors like that, you can cause the protein to unravel. You know that heat by itself will have a negative effect on hydrogen bonds, and pH and salinity can also have negative effects on those. And so when a protein unfolds or loses its native structure, that's called denaturation. And when the protein is denatured, it becomes inactive. It will not work properly. An example of a denatured protein is a cooked egg white. Egg white is almost pure protein. It's almost pure albumin. And when it's in its liquid form, it's colorless and viscous, and when you cook it, it becomes white and opaque and very different. You can't take it back again. So denatured proteins basically become inactive and are no longer useful. Now, there is protein folding that occurs in the cell, and you can't always predict what the structure is from its primary structure because there are several different stages that they go through on their way to becoming stable. A lot of these changes occur in the endoplasmic reticulum and in the Golgi apparatus. That's where those proteins are packaged and folded and formed into their finer, but the subunits are put together and so forth. Some of the molecules involved are called chaperonins, and they assist the proper folding of various protein molecules. They're proteins themselves. And we have a number of conditions like Alzheimer's and Parkinson's that are associated with misfolded proteins. And so in the case of those kind of things, something has happened to the chaperonins or other proteins that work in the folding process that make them malfunction. So part of the research that's going on nowadays about some of these conditions has to do with researching what's happening to those chaperonins and other molecules that are assisting in the folding. Nucleic acids, the final group of biomolecules we're discussing, are of course involved in the storage, transmission, and expression of hereditary information. So you know that the amino acid sequence of a polypeptide is programmed by a gene, and the genes are made of DNA. And the message telling the gene on which order to put the amino acids is coded for by the nucleotides that are the monomers of the nucleic acids. Two types of nucleic acids, deoxyribonucleic acid or DNA and ribonucleic acid or RNA. DNA is a self-replicating molecule, and it directs the synthesis of the various kinds of RNA, including messenger RNA, of course, and then through messenger RNA, it controls the synthesis of proteins, which occurs on the ribosomes, which are also made of RNA. The components of nucleic acids are called nucleotides, and so a polymer would be called a polynucleotide. Each nucleotide is made of three different things. It has a nitrogenous base, a pentose sugar, 5-carbon sugar, and one or more phosphate groups. If you look at the nucleotide without the phosphate group, it's called a nucleoside rather than a nucleotide. So if you see that term, that just tells you you're talking about the part other than the phosphate, the sugar, and the nitrogen base. So here we have a polynucleotide, or the nucleic acid. You can see the pentose sugar here. The phosphates are shown as these little yellow circles, and then the nitrogen bases are attached to the sugars. One nucleotide is composed of the phosphate group, the nitrogen base, and the pentose sugar. There are 5 different nitrogen bases divided into two categories. The two categories are pyrimidines and purines. There are 3 different pyrimidines: cytosine, thymine, and uracil. Cytosine is found in all nucleic acids, thymine is only found in DNA, and uracil is only found in RNA. All of these pyrimidines have a single ring structure. They're somewhat similar. They have slightly different side chains here. Notice that we've got an amino group on this one, and we've got a methyl group on this one, and this one doesn't have either one of those. The purines are double ring structures, and they are adenine and guanine, which are found in all RNA and DNA. Notice that adenine has this amino group here, whereas guanine, instead of the amino group, has a carbonyl group. The two different kinds of sugars found in nucleic acids are deoxyribose, which is found in DNA, and ribose, which is found in RNA. The difference is this little hydroxyl group right here on the ribose loses an oxygen to become deoxyribose. So you should be able to figure that out from the name. Deoxyribose means one less oxygen. When the polymer forms, it forms between the sugar of one nucleotide and the phosphate of the next. They're joined in covalent bonds between the hydroxyl group on one nucleotide and the phosphate on the next. Notice the designation 3' and 5'. That talks about the numbers of the carbons. When we talked about sugars, we talked about the fact that in a sugar molecule, we've got the oxygen as part of the ring, and we start numbering the carbons clockwise as we move away from the ring. So this is carbon number 1, 2, 3, 4, and the 5th one is here on the side chain. So the 3' carbon would be number 1, 2, 3, the 3rd carbon here, and the 5 carbon would be the one on the side chain. So these create the backbone of sugar phosphate units, and then the nitrogen bases are the appendages. And of course, in the case of DNA, those nitrogen bases, paired nitrogen bases, form the rungs of the last ladder. The sequence of bases is unique for each gene, and so that's a really important thing. The gene, remember, is the sequence of bases that codes for a particular protein. The RNA molecules are usually single chains, whereas the DNA molecules are double, and generally speaking, the DNA molecules are spiraling around, forming a double helix. In the helix, the backbones run in opposite directions from each other. This is called anti-parallel. So one side has the 3' end at the bottom and the 5' end at the top, and the other chain is the opposite, with the 3' at the top and the 5' at the bottom. So they're anti-parallel to each other. One DNA molecule has thousands of nucleotides, and it includes many different genes within the molecule. The nitrogen bases in DNA pair up and form hydrogen bonds between the pairs of bases. Adenine, which is a purine, always pairs with thymine, which is a pyrimidine. And guanine, purine, always pairs with cytosine, with the pyrimidine. This is called complementary base pairing. It can also occur between two RNA molecules or between parts of the same molecule. But remember, in RNA, thymine is replaced by uracil. So A pairs with U in RNA rather than A pairing with T. So here we have our DNA double helix. Notice the two antiparallel sides, 3 to 5 on one side, 5 to 3 on the other. We have our base pairs that are joined by hydrogen bonding, and that's what holds together the two sides of the double helix. Hydrogen bonding really important there. Transfer RNA is generally a single strand, but parts of it are paired up. You can see it's in this kind of twisted form here. And one end is going to have these 3 unpaired bases that will serve as the codon on the or the anticodon rather on the transfer RNA. But when you have the parts that are paired up there, you again have hydrogen bonding holding the two sides of the chain together. Now, the linear sequences of nucleotides, the sequences in a row, are passed on from parents to offspring. And when you look at related species, you're going to see more similarities in the DNA than you see in more distantly related species. And so we can use that to figure out evolutionary kinship. And so we'll talk more about that as we look at DNA later on and as we look at kinship or relationships between different groups of organisms. And that's all we have about proteins and DNA.